Hey, Dr. DiMaio here again. Hope you can hear me well. I am going to narrate your brand new Cell PowerPoints that I just made. I had to redo all the images because the images would not be allowed to make a video. In other words, my images, I guess, were so old or copyrighted that I could not use those images. So I had to redo everything. It's to your benefit. So I'm going to narrate that Cell chapter for you, or the Cell PowerPoint presentation that I have. It's similar to the one that I had already uploaded, except this is updated and a little better, I believe. So on this page right here, it's the introduction with the best images I can pull out for really grasping the cell. Is you have the capillary network with the lymphatic vessels here. You can see the lymphatic capillaries. It's identifying arterial side in red and purplish, and the venule side in the blue, and then the green is representing the lymphatic capillaries, and those little cells are the cells in between, the tissue cells. Now this is a two-dimensional image of a three-dimensional thing, so it's hard to really grasp it. In between the spaces would be interstitial fluid, and the fluid inside the cell would be the intracellular fluid, and the fluid inside the vessels of the arterioles and the venules is the plasma. And then the lymphatic capillaries are going to be picking up excess fluids that are in that system and bringing it back to the heart. And on the way back to the heart, it's going to filter them and has specialized cells. All three of these vessels have squamous epithelial cells, but in the lymphatic vessels, they overlap. And they can open like a little doggy door or a pet door, and actually fluids can rush into the capillary, the lymphatic capillary. And notice they're closed. They have closed ends. It's not a continuous loop. The lymphatic vessels are closed at one end, and they're going to bring all that fluid in one direction back to the heart. And on its route, they're going to go through an elaborate filtering system, and in that filtering system will be all your white blood cells and immune cells that are going to code for what was ever in that plasma or interstitial fluid, that may not have been good for you. To the right of the image, you see the cell membrane. It's just a different image similar to the one in our book, but it shows you the cell there, and then it shows you the membrane surrounding it, and then you're pulling it out and seeing it magnified. And you can see the carbohydrates coming off the cell membrane, which are very important for cell identity marking. Another name for cell identity marking is MHC, major histocompatibility, and it's all about that, man. It's all about your cells identifying a bad guy versus a good guy. Like I always put these analogies, you're on guard duty, halt, who goes there? Let me see who you are. And basically a cell's going to read that sugar, those carbohydrates. And that's key to immunity. And when those carbohydrates break down or get damaged through oxidative stress, your cells don't recognize it, and it starts causing cell damage. And the cell damage produces lots of mucus, and mucus can create a lot of problems if it's not dealt with. And we looked at the connective tissue proper in our lab, and we saw we have special cells that are designed to pick up excess exudate, as we're going to call it. Exudate is that material that's left over from dead cells, and cleaning up the area, the debris, getting rid of the cellular debris. So the cell, we're talking about capillary exchange, the cell membrane and membrane physiology, the organelles, protein synthesis and intercellular connections, and a little bit about mitosis. So those nodal cells are kind of like the pacemaker in your heart. You have a SA node and an AV node, the two pacemakers of the heart. One gets the atrium to beat, and the other one gets the ventricles to beat. This is going to cause constriction around a little tiny capillary sphincter, precapillary sphincter, and the nodal cells will blip on a timing basis, and they'll constrict and build up pressure from the arterial side. And then as the pressure builds up, it could push fluids out of the capillary. Some of the plasma gets pushed out, and it ensures two things: that you get a nice exchange from those capillaries, but also, if you notice, there's the meta-arterial or metarial, whichever way you say. I say meta-arterial, and that's also known as a thoroughfare. and that is allowing blood to make sure it goes from arterial side to venule side. As I always talk about pressures, being a hydraulics mech on jets in the Marines, the heart pushes out the fluid from the aorta at 80 psi, 80 pounds per square inch. By the time it comes to this precapillary bed here, it's a lot lower. And then as it comes out past that, the venule, it's much lower. So we're talking like, I think 36 or somewhere around there, psi at the arterial side, and then only like 15 psi, I think, on the venule side. It could be wrong, but there's a drastic difference. There's a big difference from when it came out of the aorta and from when it's going to the venules back to the heart. So you want to make sure you have a nice thoroughfare to make sure everybody gets back to the heart. And then you had those precapillary sphincters, we had those lymphatic vessels all throughout that's not shown here, and that are capturing the excess plasma that's lost on every cardiac cycle. That means every time your heart beats, you lose plasma. If you didn't recapture it, your cardiovascular system would collapse. So the lymphatic system has to recapture it and bring it back to the heart. So capillary exchange, you're going to have arterioles, and you can see here on the image the arterial side. Blood's coming into the bed, and the meta-arterioles, it has a thoroughfare bringing it all the way through. It's going to go to the venule side. It doesn't really show you the thoroughfare too well here. And then you have the precapillary sphincters. I don't believe they're showing it here that well either. And then the lymphatic capillaries, which is nice about this image. You see everybody together, except for the interstitial fluid. You can't see the fluid. And this is a reality about the capillary bed in your body. So when you take gross anatomy in like a higher level, like I did in college, and if you go on to a higher degree, you may be doing it yourself. And things are changing in that area because now they have these new computerized tables. But when you do dissection or you're trying to memorize stuff, we used to use a multicolor pen, and red was artery, blue was vein, green was lymphatic, and yellow was nerve. So when you do gross anatomy and you look at a cadaver, you say, okay, let's identify the artery, the vein, the lymph, and the nerve for that part of the body. Always trying to trace that. You always try to keep that intact when you cut. And if you don't keep it intact, then you lose it and you can't tell. And when you take a test in a cadaver lab, you have to identify the artery, the vein, the nerve, and the lymph. And it gets more specific as you go on at higher levels. But you can see how this is a standard in anatomy. Blood supply, nerve supply, and lymph supply. So now we're going to have an exchange going on here. The products that are in the plasma diffuse out from the capillaries. Now the plasma is the liquid portion of blood, right? And so it's going to diffuse out of the capillaries into the spaces, the interstitial space. That's the space in between all those cells and vessels. And then the interstitial fluid is going to have both the products from the plasma, but also good and bad products that came out of the cell. So the cell may have made a hormone. That's a good hormone that you need. That would be in that space. Or the cell could have been releasing, urea and CO2, which is stuff that has to be removed. So that stuff is found in that space there, both the good and bad from the blood, both the good and the bad from the cells. And then the cells will take in the fluids and based upon diffusion. So if there's not enough oxygen inside the cell, that oxygen that diffused out of the plasma into the interstitial spaces will come into the cell, right? And if the cell had released CO2, that CO2 would go into the venous system and the lymphatic system, let's say. So, and then the waste will come out and go into those systems as well. And then the lymph will carry all the bad stuff. It has specialized squamous epithelial cells that can snap open because they're overlapped. And when the pressure outside is strong enough, it pushes the door open. And then that fluid comes into the lymphatic capillary and snaps back shut. And it's closed at one end, so it's either going to go to that end and stop, or it's got to go back up. And this is why in healthcare you want to get people moving after surgery. You don't want them to be in bed too long. and they use those pressure cuffs to push fluid along. You want to keep that fluid moving along because there's no pump for the lymphatic system. The lymphatic system moves the fluids up towards the heart through gravity and muscle contraction and muscle relaxation. And that's why it's so important to move and exercise. All right, so cells will receive their nutrients from the blood. Remember, the blood's traveling through the capillaries, and the capillary bed is where exchange occurs. The exchange is mediated by the interstitial fluid where it surrounds the cells, and the materials can diffuse across cell membranes, across membranes of the capillaries and cell membranes. The lymph capillaries can take up excess materials and also bring them back to the heart. In doing so, the lymph nodes will code for bacteria, virus, fungi, and cancer. Yep, we're always doing that. Okay, so cells have many shapes and sizes. Cells are the basic structural functional unit of the body, sir, as atoms are the building blocks for molecules. Cells are the building blocks for the body. Cells are the smallest functional units of life, the structural functional unit of the body, and each cell maintains its own homeostasis, and they work together for the good for the whole, just like you. Okay, cell diversity. This is showing you different shapes, a little different image from our book. Up top on the left, it's showing you muscle, and in the center, it's showing you like a stem cell that could become anything. To the left is a blood cell. To the bottom left would be a neuron, and that would be an anaxonal neuron. There's no axon, it's just dendrites on that one. And the bottom is a cardiac muscle cell. And to the right looks like a blood cell, and to the top, like a white blood cell or any other cell. It's hard to tell what that cell is supposed to be right there. Could be a cuboidal epithelial cell. Up top would be a cell with either microvilli or cilia. It's hard to tell what they're trying to represent there. But cells have different shapes. That's the slide. And all the parts of the generalized animal cell are here. This has the cilia on it, this one. So it's not microvilli there. So on the bottom, you see number 8 is the cilia in this one. And you have all your organelles, especially the nucleus. What is the only anucleated cell in the body? Do you remember? What's the only anucleated cell? And anucleated means does not have its own nucleus. That would be the red blood cell, otherwise known as what? Erythrocyte. And if you don't have a nucleus, you're going to die. You're not going to last long. Because the nucleus is extremely important because it has the DNA that tells everything what to do in the cell, including the repair of the cell. So cells can't repair without DNA. And as we're going to see at the end of this chapter, we're going to talk about protein synthesis in that we use DNA to drive it. RNA is important, and there's 3 types of RNA involved in this protein synthesis. There's messenger RNA, there's ribosomal RNA, and there's transfer RNA. And it's going to be a process of making a copy of a gene from the DNA in that nucleus for whatever it needs to make. So let's say the cell here is going to make insulin. The copy has to come off of the gene. The gene has a series of codes on it. Those codes are transcribed as mRNA. That's called transcription. When you transcribe it, it's going to place it on a table called the ribosome. And the ribosome is basically ribosomal RNA or rRNA. It's like a table. And it is the site of protein synthesis. And then transfer RNA is going to be bringing the specific amino acids necessary to build that amino acid. So inside that nucleus, you have your DNA. Number 15 is showing it as chromatin. Why is it chromatin, not chromatids or anything else? Because it's at rest. This is an interphase right now. This cell is in its interphase stage. It's not going through cell replication yet. But you see, all that protein synthesis could be doing, the protein synthesis we just talked about was his job to make insulin, but also he has to help maintain his own homeostasis, and he has to repair his cell membrane and organelles and so on. So not only is protein synthesis occurring, so is carbohydrate synthesis, and so is lipid synthesis. So wherever you see Endoplasmic reticulum, that's where those three things occur. If there's ribosomes on it, they call it the rough ER. So number 5 is showing it as rough ER. You see on the nucleus also, you see these nucleopores, and the nuclear envelope has these nucleopores. Number 2 is the nuclear envelope. They don't really show nucleopores, but you see those little connexons, or those little deuterus, allowing things to pass through, the little yellow guys there. And then inside number 16 is showing you that's the nucleolus, which is the site where ribosomes come from. So ribosomes are first formed within the nucleolus. And then the endoplasmic reticulum is always attached to that nuclear envelope. And you can see rough ER here. And that would be labeled as number 5, I believe. And the smooth ER is labeled as number 14. Notice smooth does not have ribosomes, rough does. And then the mitochondria is number 7. Number 10 would be the centrioles within its centrosome. It doesn't show the centrosome, which is like where they're sitting on top of. And notice they're at rest right now. That's not really doing any job of mitosis right now. And then you have like peroxisomes and lysosomes. Number 11 is representing a lysosome on this model, and number 6 represents a peroxisome. And what else we got here? So the cilia. Cilia was number 8. Number 9 is the cytoskeleton, I believe, intermediate filaments of the cytoskeleton. And number 13 is just representing the cytosol, which is the fluid portion and non-membranous stuff. Number 12 shows you some free ribosomes. So you have fixed and free. Okay, let's go on. So now, the plasma membrane, sometimes is called the cell membrane or plasma membrane, has 4 basic functions. One, of course, is a physical barrier, creating physical isolation of the cell from the inside versus the outside. And remember, inside the cell is different than the outside, and inside has more water. And we'll talk about the membrane charge inside is different from the outside. It regulates exchange, the cell membrane. It allows certain things in and certain out. It has sensitivity. It has receptors, and it can respond to different things. And it has structural support, cell connections that connect cells to each other. So the cell membrane has been termed to be a selective, semi-permeable, phospholipid bilayer with proteins and carbohydrates. Now, selective allows certain things in, certain things out. Semi-permeable does allow stuff to just pass right through the membrane. And it has a phospholipid bilayer, which is important, helps to seal it. It also protects it. And it also has proteins lodged in there and carbohydrates. Sometimes they call the membrane, the way it looks, is a fluid mosaic model, meaning it looks like a mosaic with things stuck in there, but it's fluid and it moves around. Now, I did tell you, and I'll say it again, that some textbooks have said that those phospholipids can flip position up to almost 1,000,000 times per second, which is phenomenal. Now, the phospholipids and other lipids make up about 45% of the total weight of the cell membrane. And the phospholipids are hydrophobic and hydrophilic. So one side loves water, one side hates water. Which one loves water? Philadelphia is the city of brotherly love, so hydrophilic means it loves water. So that's the phosphate heads. And the fatty acid tails are hydrophobic. When I say 45% by weight, you got to realize the lipids take up most of the area around it, but by weight, it's heavier in protein, number 2. And proteins make up about 50% of the weight of the cell membrane, and they can be integral, that means within the entire cell membrane, and they could be transmembrane, meaning they could pass through both sides, and they have some on the surface, on the outer surface, some on the inner surface. They could be surface membrane proteins. Each protein has a very specific function, which we will go over. And carbohydrates have specific functions as well. They make up about 5% of the weight, and very important for cell identity marking. And you see here in this image, You see the carbohydrates coming off the proteins, the glycoproteins. That is your MHC, major histocompatibility. That's what identifies you as you. Only you have that shape, okay? And so whenever a cell is going to identify an enemy, he's going to look at that carbohydrate coming off the protein. The glycoprotein is the cell identity marker of the cell, otherwise known as MHC, major histocompatibility. And then you can see these proteins embedded in there. Some have a function of being receptors. Some are going to be functioning as ion channels, as we'll see. Here's the functions of membrane proteins, all on one page. On the left at the top, some are transporters allowing ions to flow through ion channels. That could be like a leakage ion channel. Next to it, to the right, is enzyme activity. So sometimes they work within what's called second messenger system inside the cell, or just function as enzymes. All the way to the right on the top, some have receptors, surface membrane receptors. And remember, we talked about certain hormones. Some cells in our body, all the cells in our body have receptors for certain hormones, like thyroid hormone. Okay, and then bottom left, the cell surface identity marker, we talked about that. That would be a glycoprotein with the sugar coming off the protein. Cell adhesion molecules, where they actually create spot welds or connections to other cells. And then the bottom is attachment to the cytoskeleton. Again, here we just see some of the attachments to the cytoskeleton. We have the different ways the cell can tightly attach to each other, and they call them membrane tight junctions. If you look there, we have a desmosome, and we have gap junctions. Okay, and a gap junction is unique in that you can see on the bottom right, it has connexons, which allow ions to flow through both cells, and cells can communicate like that. And gap junctions are very prevalent, extremely prevalent in cardiac muscle cells. Once one cardiac muscle cell contracts, the next one does because of that, because calcium ions can flow directly from one cell to the next. So we have different ways things get in and out of the cell, and diffusion through a plasma membrane. So all the way to the left is simple diffusion. Some lipid-soluble solutes can pass right through that phospholipid membrane. And let's give you an example of a couple. Number one is hormones, such as sex hormones. That would be estrogen and testosterone, and also cortisone can pass directly through the cell membrane. And then a very bad thing is benzene gas can pass right through the cell membrane, which is carcinogenic. Next one over shows you large carrier-mediated facilitated diffusion, in that this large protein is specific for a specific chemical that can fit into a shape. When it does that, it goes through a shape change or transformational change, and it pops it in. It doesn't need any ATP. All of these things here are passive. That means they don't use ATP. They don't need energy. Just a concentration gradient. And then the channel-mediated ion facilitated fusion, you have protein ion channels allowing charged particles that could not pass directly through the cell membrane because the phosphates have a charge. So they go through these ion channels. And then the last one is for osmosis, the movement of water. And water can pass right through a cell membrane to a certain degree. And some proteins are actually formed into aquaporins. Okay, now active transport is different. Active transport means what? It means it needs ATP to run. So the sodium-potassium pump all the way to the left is called primary active transport. And the unique thing about that is that it pumps out, it doesn't show it here, but it shows it in our other notes, it pumps out 3 sodium for every 2 potassium back in. And that creates a concentration gradient of sodium being more sodium outside. So that's a chemical gradient of more sodium outside and a chemical gradient of more potassium inside. It also, because they both have positive charges, but you're losing 3 and only gaining 2 back, it creates an electrical gradient. So inside is going to be less positive just from that alone. And we'll talk about more reasons why inside is going to be even less positive. to the right, it looks at another type of active transport, and they're calling this secondary active transport. And that is when the glucose comes in, the sodium comes in passively, like a little birdie landing on your shoulder going into a building. The door was open for you already, let's say, and it lands on your shoulder, and it's coming in passively on your shoulder. And now you have to work at getting that sodium out. So it goes back through primary active transport to get it out. So they call it secondary active transport because the sodium came in passively and is pumped out actively. Other types of active transport include vesicular transport, such as endocytosis. Under endocytosis, you have phagocytosis, which is cell eating, pinocytosis, like Pinot Grigio, cell drinking, and receptor-mediated endocytosis, where it has to be connected to a receptor first and be pulled into a vesicle. Now, it can work both ways. You can have endocytosis, where stuff comes in, looking on the right, and you can have exocytosis, where a cell releases products. All require ATP. So on the left shows you passive, on the right shows you active. On the left does not require ATP, on the right does require ATP. So let's look at passive. It's a review. Simple diffusion, which includes osmosis, protein ion channel mediated diffusion, and protein carrier mediated diffusion. On the right, active transport, you have primary active transport, the sodium-potassium pump. Secondary active transport, sodium came in passively like a little birdie on your shoulder, and then it gets pumped out actively through the sodium-potassium pump. And then vesicular transport. We had the different types of vesicular transport. You had endocytosis and exocytosis. Under endocytosis, there were several different types. Now, diffusion has to do with how things move from an area to another area. So diffusion is defined as, you got a pen, right? How do you define diffusion? The random mixing of particles from the area of high concentration to low. I'll say it again. Diffusion is the random mixing of particles from an area of high concentration to low concentration. When we say high concentration of solute to low concentration of solute. So here they're putting something in a beaker. Looks like it's just a dye, and you see it just initially go in, and it's slowly randomly mixing until it comes to what's called equilibrium. So diffusion is based upon concentration gradient, and there has to be a time of that to come to equilibrium. So sometimes, the amount of the product and the time you give it tells us what it's going to do when you deal with diffusion, right? Osmosis is very similar to diffusion, but it's like the diffusion of water. So it's kind of like we were talking about particles in diffusion going from an area of high concentration to low particle, low solute. Now with water, water tends to want to move the opposite way. And water, if you look on the left side, you have water as the solvent, and those little purple or blue dots are the actual solute that's in that water, and there's a membrane in the middle. And so the water is poured in there initially, and then it moves through the membrane to go from an area of low solute to higher solute. On the right, you can see a lot more particles. And so it increases, there's more pressure on the right because there's more fluid and more solute. So osmosis, it's a phenomenon of flow of liquid through a semi-permeable membrane without any external force. You've heard they say water always wants to seek its level. So you can have different concentrations of solute in a solution. If you remember, I think I gave you this definition: a solvent, a solution, and a solute. Okay, so first of all, let's talk about the solvent. The solvent is the thing, the fluid that's going to have stuff dissolved in it. So the solvent for the human body is water. The human body's universal solvent is water. And then we'll have stuff dissolved in it. If you have sodium ions dissolved in it, that's the sodium ions are the solute. And then together they form a solution. So if the human body has a certain number of sodium ions normally in our solution, our plasma, and you put a red blood cell in a beaker that had the same amount of sodium ions, let's say, and other ions, that beaker of fluid would be considered to be an isotonic solution. The same. Iso means the same. If you have a hypotonic solution, that means you have less solute. Hypotone, less solute. That means more water. If you have a hypertonic solution, that means you have a lot more solute than water than normal. Okay, so here shows you red blood cells in a isotonic solution. The amount of water transported into the cell is equal to the amount that's transported out. So the solute concentration in the cell is equal to the solution. No exchange really, no change to the cell. The cells stay healthy in that type of solution. So when you give a patient fluids, you want to make sure those fluids are isotonic, right, for health. And then hypotonic solution means that the cells are going to have a lot more solute within the cell normally. And what's going to happen is if you put a cell, like a red blood cell, in pure crystal water with nothing else, that water has nothing else in it, meaning distilled water, there's no solutes in it, the cell will actually burst because the water is going to rush in to try and find its equilibrium. In other words, we said that water moves from area low solute to high solute. So the water in the beaker was low solute. It's going to enter into the cell that had more solutes. And so the cell would burst. If you cell in a hypertonic solution, meaning lots of salt, a salty water that has more salt than the cells you don't normally do, the water inside the cells are going to seek its level and leave the cell and go into the higher solute. And then that cell will shrink and crenate, they say, and die, basically. So here's the three together. Hypertonic means the water is going to leave the cell and go into solution because the solution was so much solute in that solution that the cells would crenate. Isotonic, same, they're happy. Hypotonic, water is going to rush in and cause the cell to burst. Filtration is a passive process, and we have filtration going on in our body when we filtrate into different things, especially the kidney is a good example of filtration. And the hydrostatic pressure forces the water movement, and the blood pressure causes filtration in small vessels. So diffusion, in reality, in the body, we have protein ion channels that allow for small inorganic ions to pass through. This is generally slower than simple diffusion, where you can just have things pass right through the phospholipids. The area they move through is smaller than the entire membrane, but the need is there because of the charge, right? Although it is smaller and slower, it actually moves pretty fast, and about 1 million potassium ions per second can move through those ion channels. Osmosis and osmotic pressure, or hydrostatic pressure. So we have this opposing pressure to water, and that would be osmotic pressure. So you can have water build up, fluid is running, or you turn the hose on, small amount, there's very little water running, it's low pressure. The more water, the higher pressure, and the more you resist it, the higher pressure. So if you put your finger at the end of the hose, you resist the pressure, that's called osmotic pressure. That's the opposing pressure. If you could take the entire hose and squeeze it all the way around, it would actually increase the pressure. And that's what happens in our body. Our blood pressure can be elevated from muscles around those arterioles we saw in the arteries that can be squeezed and increase blood pressure. And then they could dilate too to relax it. And so when you have resistance, you can increase pressure. When you reduce the resistance, you lower the. So like I said, protein ions allow for small charged particles to pass through the pore of a transmembrane protein. It could be passive or active, meaning you can actually trigger it to pass through by opening up a gate. So we have different types of ion channels. Okay, so Ion channels can be leakage. That's the first one you see there. And it randomly opens and allows things to pass through. Or they can be ligand gated, meaning a chemical, like a neurotransmitter or a hormone, fits into a receptor associated with the ion channel, and then the gate opens and allows a flow of specific ions through there. Some are specific for like sodium or potassium or calcium. And then you have mechanically gated ion channels that are opening and closing in response to a mechanical stimulus, like a stretch or pressure. And then the voltage-gated ion channels are opening and closing in response to a change in the membrane potential. So that brings us to membrane potentials. So membrane potentials, meaning that you have a difference in the charge inside versus the outside. And there's resting membrane potentials, graded potentials, and action potentials. So all cells have a resting membrane potential. It is the difference of charge inside versus the outside. Most cells are measured at rest at minus 70 millivolts, meaning inside is more negative than the outside. And I taught you last time, I believe, that a millivolt is 0.001 volts. Very small charge. Only neurons have all three. They have resting membrane potentials, they have what's called a graded potential, and they have action potentials. Now, a graded potential is varying in amplitude. It doesn't go only one amplitude. It can vary. And if you think about graded potentials, you could think about you get a change in the membrane potential from its normal rest, and it starts depolarizing. Ions start rushing in. But it could travel a short distance and be very weak, or travel further and be very strong, and it can go back. So that's a graded potential. An action potential is an all or none. Once it starts depolarizing the membrane and ions rush in, it's going to travel all the way or not. But neurons can have all three. Muscles have both resting membrane potentials, and they can have action potentials. And the action potential is what triggers the muscle to contract. And there are only two types of excitable cells in the body, and that would be neurons and muscles. The cytosol is the fluid portion of your cytoplasm. It also it also is associated with the non-membranous organelles within the cell. And organelles are usually the cytoplasm that surrounds the organelles is composed of 70 to 90% water. Remember what I said, there should be more water inside our cells than outside in a healthy individual. Inside our cell there will be dissolved substances as well, suspended compounds, amino acids, carbohydrates, lipids. It is the site of many chemical reactions, and enzymes catalyze these reactions, resulting in energy produced. That energy is captured and drives all the cell's activities. So again, on organelles, there's specialized structure that performs specific functions. The number and type of organelles differ per type of cell. It depends on the overall function of the cell. Non-membranous organelles lack a membrane, such as the cytoskeleton, centrosomes, cilia, ribosomes. And membranous organelles are surrounded by one or two membranes, such as the endoplasmic reticulum, the Golgi complex, the mitochondria, or the nucleus. Cell membrane modification. Some cells have modifications. We talked about cilia and microvilli. Here's an example of cells with cilia and microvilli. And you can see it attached to the cell, the basement membrane at the bottom is a little lamina and basement attaching to the connective tissue below. And then you can see up top, on the top of the surface, microvilli on the one to the right and cilia on the one to the left. Now, ribosomes are tiny organelles made up of just basically ribosomal RNA. They are the site for protein synthesis in the cell. You have both free ribosomes in the cytosol and membrane-bound ribosomes found in the rough endoplasmic reticulum. So here's an example of free versus bound. Do you remember the difference? The difference is free ribosomes synthesize proteins for use within the cell, and the bound ribosomes synthesize proteins for export, or sometimes to use in making lysosomes. Lysosomes help digest bacteria. Okay, so endoplasmic reticulum is a network of membranes that's found throughout the cell and attached to the nuclear membrane. The rough ER, or rough endoplasmic reticulum, has ribosomes associated with protein synthesis. The smooth ER does not have ribosomes, and that's associated with the synthesis of phospholipids and other fats and steroids, and also carbohydrates. And so here you see the endoplasmic reticulum is always attached to the nuclear membrane, and it's showing you both rough and smooth here. And the four functions of the endoplasmic reticulum are synthesis of macromolecules, storage of molecules, transport of molecules, and detoxification. The Golgi apparatus, this is a good image. It's kind of showing you how the Golgi apparatus is interacting. It's a series of flattened membrane discs that give rise to lysosomes, secretory vesicles, and membrane renewal vesicles. You can see the Golgi apparatus on the right there in the image has these sacs filled. They're actually called cisterns. And then they reprocess what was made by the endoplasmic reticulum, and they can either ship it out as a transport vesicle or a secretory vesicle, or it can actually make lysosomes as well. And so here's another image of the relationship between the ER and the Golgi apparatus. You see number one showing a protein-containing vesicle pinching off the ER, which migrates and fuses with the Golgi apparatus. And then the proteins are modified within the Golgi compartments. And the proteins are then repackaged into different vesicle types depending on their ultimate destination. Secretory, or lysosome, or membrane renewal type of thing. Okay, so peroxisomes that are made by that contain enzymes that oxidize organic substances. Lysosomes have enzymes that can recycle structures of the cell or foreign substances. The term autophagy is the process in which a worn-out cell organelle is digested and recycled by lysosomes. Autolysis is when the lysosomes destroy the entire cell and self-destruct. The mitochondria, the powerhouse of the cell producing ATP, has its own DNA, and we inherit that from our mother. So if you look at the functions on the left, it's the most important function is to produce energy. It takes the molecules from nutrition and processes them and creates ATP. And that process is called oxidative phosphorylation, or you could just say phosphorylation. Mitochondria help cells to maintain proper concentration of calcium ions as well. And the mitochondria also help building certain parts of blood and hormones like testosterone and estrogen. And the liver cells, the mitochondria have enzymes that detoxify ammonia. Here's a little example of mitochondria, and it has its own DNA in there. And you're seeing fat and sugar intermediates go through a series of processes, and it makes different things. It's going to make ATP, number one. That's the most important thing. So here's the nucleus. The nucleus has a nuclear envelope with nucleopores. Within the nucleus is the chromatin, or chromosomes, and chromosomes start forming there, and there's chromatin free form there. And then you have a nucleolus. So the nucleolus is where the ribosomes are formed, and the nucleus is where protein synthesis is regulated. Protein synthesis is the assembling of functional polypeptides in the cytoplasm. It involves gene activation, the uncoiling of a DNA and temporarily removing histones from it, and then transcription. Transcription is the synthesis of RNA from DNA template. And all RNA, including messenger RNA, is formed through transcription of DNA. So now the first step in protein synthesis really is activating the gene and then transcribing the gene onto a template called messenger RNA. Now, you're going to have two strands of DNA normally. Coding strand specifies the sequence of amino acid polypeptides, and the template strand is used for the mRNA. So in other words, what they're saying here is when you look at a DNA strand, you're going to take a coding strand to make that a specific protein. So it's a series of nucleotides along this long gene. And then the mRNA is going to make a copy, a template strand of that. But when the mRNA makes it, is uracil, replaces thymine for uracil. We talked about that. So translation is different. So you have transcription is the first thing, and then translation. So after leaving the nucleus, the mRNA binds to the ribosome. It gets on the table. So it's a pattern on the table. And each mRNA have a codon. The codon is a triplet pair, a triplet of nucleotides. And each one of those codons or triplets of nucleotides translates to a specific amino acid. And those specific amino acids are delivered by another RNA called tRNA or transfer RNA. And on the tRNA, it has what's called an anticodon, which is the complement base pairs for those triplets of the codon. And it's complementary to the mRNA's codon. And now enzymes join amino acids together with peptide bonds, and then a stop codon stops it. So look at this image here, if you can make it out. All the way to the left is the actual nucleus and the nucleopores. You see that? Number one, the nucleus serves as a template, the DNA serves as a template, and then the mRNA makes a copy of that. And then it leaves the nucleus. So that number three is showing when the mRNA is formed, it has codons on it. You can't see it here, but it has the three codons on it. And then the mRNA moves to the cytoplasm and becomes associated with a ribosome. And so it goes on the table, the ribosomal table. So number five, the tRNA with its anticodon carries an amino acid to the mRNA. And that's going to actually lock in, and it's going to create a protein using peptide bonds, number 7. And then number 8, the tRNA departs, and we'll soon pick up another amino acid. Let's see another. So what is a codon? What's an anticodon? So the codon is 3 nucleotides in the mRNA. Each codon will code for a single amino acid. So look at the bottom. You see the mRNA strand with the CUC, cytosine, uracil, and cytosine. That's 3 nucleotides. That's the codon. And then you see the green guy there, the anticodon, has 3 nucleotides as well, and that is the opposite or the complement base pairs for those, the codon. So they call it an anticodon. Each codon and anticodon is representative of a specific amino acid. So what you're seeing here is leucine, LEU, is attaching to the tRNA. So that green guy is the tRNA with its anticodon. And then he's going to attach to the mRNA's codon. And then another amino acid is going to come along and it's going to assemble the protein. Here's a good example. On the bottom, you see the mRNA strand, the bottom left side. And then you see they have codons, like the AUG and the UUC. Those are 2 different codons. The AUG codon is representative of methionine. That's an amino acid. The UUC codon is representative of phenylalanine. And so you see the tRNA looks like a T there. He's going to grab its amino acid associated with the anticodon and codon that he has. The tRNA has the anticodon for the codon. So remember we said that if you have one strand of DNA, you can always tell the other strand because they have the complement base pairs. They'll always match. Same thing with codon and anticodon, they will always match. And so this is how proteins are synthesized, through transcription. You're taking that pattern as the mRNA for the gene to make whatever it is. And translation is bringing the amino acids to the table and fixing it to and locking in on the mRNA. And then at the top, the tRNA is actually going to have peptide bonds bonding each amino acid. So the table is the ribosomal RNA that they don't label it here, but on the bottom, the brown thing that's kind of coming down there like a glob, that's going to be part of the ribosome. And that's where the mRNA is going to be sitting on. And then the tRNA is going to be grabbing amino acids from the cytoplasm from cell eating and from capillary exchange. And then it's going to be bringing it in, and it's going to create these bonds there. Quite amazing. And so there's three different types of RNA associated with this protein synthesis. One is messenger RNA. Two is ribosomal RNA, which is a ribosome, by the way. And three is transfer RNA. Cell cycle. The cell has a cycle. It has a growth cycle. It has a cycle when it's making new DNA. It has a growth and preparation for mitosis. And then there's steps or different phases of mitosis called cell division. So before you even go into mitosis, you have to think of the growth stage as G1. The DNA synthesis is the S phase. And the growth and preparation for mitosis is G2. The M is representing mitosis, the different phases of mitosis. So here's another cool picture that kind of shows it all. At the bottom left there, you can see the S, the G1, the S, and the G2 phase. And in the G2 phase, you can see that the sister chromatids are starting to become loosely coiled. And then in prophase, the spindle fibers appear. In pro-metaphase, you have spindle fibers attached to the chromosome. Then metaphase, you have the chromosomes aligned. Anaphase, sister chromatids move to opposite poles. And the telophase is when you start getting a separation with a cleavage furrow, and the nuclear membrane forms around the new cell's new nucleus that's being formed. And then cytokinesis is the complete separation of the two sister cells or daughter cells. That's a really good image to help you to understand it there. And then here, to summarize it all, in cell cycle, you have an interphase, which is including the G1, S, and G2. So that's at rest. We think it's at rest, but things are really happening, like we said. In G1, we talked about growth and doubling of the organelles. And in the S phase, DNA replication is occurring. And in the G2 phase, growth and preparation for mitosis occurs. And the basic mitosis phases are prophase, metaphase, anaphase, and telophase. And I know some books, and including ours, and also the lab, has a early and a late stages of each one of those. But basically speaking, you have the 4 phases of mitosis. Alrighty, thank you.